Hello, I'm Vincent, a PhD student in geology at Trinity College Dublin. My research project lies on the study of the chemistry of biomineralizations to reconstruct past climates at a high temporal resolution. In climate prediction, scientists need information on how the climate was in previous times in order to model the future. Part of this data is obtained by the study of the chemistry of sediments, slowly deposited on top of previous strata. But this method is useful only when we want to predict climate at a large timescale. My work focuses on the study of biominerals, which are all hard parts synthesized by organisms. These include bones, teeth, shells, coral reefs, and many more that can be found as fossils. These structures are built all along the life of the individual, with elements it samples from its environment. Thus, by investigating biominerals, we can see the seasonal evolution of the environment throughout the life of the organism, which is generally a few years. While using sediments, each analysis corresponds to a mean of thousands of years.
We want to do two things. We want to sample the specimen, and then we want to analyze the specimen. And because we're dealing with a very slow growth of material, we want to take a very small scale sample to signify a small time interval. So there are two main ways we can take a sample. One is using a drill, and using this drill we can take a very small sample of the biomineralization. The second way we can take a sample is by using a laser to take an even smaller sample volume. So once we actually have the sample, what we want to do is then analyze its composition. And to do this, we can use an instrumentation called mass spectrometry. Now, mass spectrometers are quite sophisticated devices that allow us to measure very small concentrations. Think about a thousandth of a drop of water in an Olympic-sized swimming pool.
Unfortunately, it's not that simple. All metabolic activity, like stress or reproduction cycle, can have an influence on the mineralization, and we obtain a disturbance in the record called vital effect, as if two people were talking at the same time. You can understand neither the first nor the second very clearly. My project will focus on various biominerals, including coral skeletons, fish teeth, bivalve shells, and sea urchin spines, from deep-sea ecosystems in our rich waters, where the environment is more stable and organisms have less stressful conditions compared to the shore, to reduce the influence of vital effects. That way, we can clear the environmental part of the record and eventually provide short timescale environmental data to predictive models for climate change.